We are now going to go into more details on each of the 3 steps of cellular respiration. So first, we're going to start off with glycolysis. So let's take a look at where we left off. We're going to take a close look at the cell. So we just had the blood, and glucose was in the blood. And the glucose is going to leave the blood and enter the cell. So we're going to make this entire screen practically the cell. So the glucose is going to enter into the cell cytoplasm. And here's where we're going to do our first step, which is glycolysis. So let's do glycolysis in blue. So step 1. Glycolysis. So we're going to start with our glucose. And in addition to the glucose, in order to do glycolysis, we'll need 2 ATP. Think of these ATP as an initial investment. We're going to make a lot more ATP at the end, but this initial 2 ATP investment will get us started. We'll also need 2 NAD molecules, and I'll explain those a little more. All right. So what's going to happen is we're going to take this glucose molecule, and this glucose molecule is actually a chain with 6 carbons. So I'm simplifying the glucose, just drawing the carbons so that you can see there's 6 of them. In glycolysis, what's going to happen is that we are going to split the glucose into 2. And that's actually what the word glycolysis means. Lysis, right here, lyse, remember, means to break. And then the glyco in the beginning stands for the glucose. So glycolysis is literally breaking glucose in half. So what we're going to end up with is 2 pyruvic acid molecule. Each one is going to be 3 carbons. In addition, you'll get 4 ATP. But remember, you had to invest 2 in the beginning, so in reality, this is a net of 2 ATP. In other words, we gain 2 ATP. And we'll get this molecule called NADH, and we'll get 2 of those. All right, so let me explain the NAD and the NADH. So one important thing about the glucose is that we will be harvesting the protons and electrons from it. So in this glucose molecule, you'll have a lot of protons, which is the hydrogen without the electron, and then you'll have those electrons. And the energy from the glucose molecule is really going to be stored in these protons and electrons. And we're focusing on the electrons. So what we're going to do is we're going to put this onto the NAD. So basically we're adding a hydrogen atom onto NAD, which is how it becomes NADH. And again, it's important because we're going to be tracking these electrons. So there's electron on that hydrogen atom, and we'll be tracking those electrons to use later. Think of the electron as the energy carrier. It's carrying energy.
I like to think of NAD as similar to lift. What does lift do? It carries passengers from point A to B. In this case, the critical passenger is the electron. Remember, the electron is carrying the energy from the food you ate. So NAD, or lift, is going to pick up that electron from the glucose from the food you ate. We will see where it is going later. Remember, following the first law of thermodynamics, energy can only be transferred. In this case, NAD, our lift, is transferring energy in the electron.
This is the process of glycolysis in the presence of oxygen. So if oxygen is around, the pyruvic acid is going to continue on into the next step, which is going to be the citric acid cycle. So again, this is if there's oxygen around, or in other words, if it's aerobic. Remember, aerobic, think of air, you're breathing oxygen. Oxygen is present, so with oxygen.
The reason why oxygen is used here is that in order for glycolysis to occur, you need NAD. If there is no NAD, there is no glycolysis. NADH gets recycled back into NAD in the presence of oxygen. Without oxygen, we need an alternative way to get our NAD back. If there is no oxygen, then you can't recycle NADH back to NAD. So instead, you need to use lactic acid.
Now, a slightly different process takes place if there's no oxygen present. I'm going to go ahead and draw this in this color. So now let's look at what happens if we are dealing with anaerobic respiration. So the an means no, so this is without oxygen. So without oxygen, the pyruvic acid is actually going to turn into another molecule called lactic acid. In order to make lactic acid, the hydrogen with that electron needs to go onto the lactic acid molecule. And in this way, the NAD is remade. And so the NAD can resupply the NAD at the beginning of glycolysis. So that way we can continue having our glucose from the food we eat, the ATP and the NAD, and we can continue the cycle to keep gaining this net of 2 ATP. So again, this is without oxygen present. Pyruvic acid turns into lactic acid with the help of getting the hydrogen from the NADH, and we now have a buildup of the lactic acid, which is actually what makes your muscles hurt while you're actually exercising, because when you're exercising really hard, you have no air, and it's this lactic acid that starts to build up, which causes that sharp pain while you're exercising.